Mitosis begins in interphase and has a total of three subphases. It starts with G1, and during G1, the cell is living its normal life, doing all of the things a typical cell does. Inside of the nucleus, we have 46 chromatin. Chromatin is going to refer to uncondensed and loose chromosomes, and they look like these thin threads. After G1, the cell enters S phase, and this is where DNA replication occurs. Each of the 46 chromosomes is copied and it's going to form sister chromatids. So now the cell has 92 chromatids, but it still only has 46 chromosomes because each chromosome is going to consist of two identical sister chromatids that are held together at the centromere. So essentially, chromatids refer to the individual DNA strands that make up a duplicated chromosome. They are called sister chromatids when they are paired. The last part of interphase is G2, which isn't shown here, and during G2, the cell grows a bit more and gets ready to divide. So after G2, the cell finally enters mitosis. Mitosis starts with prophase, and during prophase, those sister chromatids, they're going to condense to form duplicated chromosomes. And what I mean is that these chromatids, they're going to coil tightly, and they're finally going to become visible under a microscope. Condensing DNA is going to keep it organized and compact, which is important for the division process. Then comes prometaphase. Not every textbook shows this as its own stage, so if yours doesn't, you can group it in with metaphase. During prometaphase, the nuclear envelope it begins to break down and our spindle fibers are going to begin to interact with chromosomes. Specifically, kinetochore microtubules are going to attach to kinetochores on each duplicated chromosome. And these microtubules, they're going to be responsible for moving those chromosomes around. Then we enter metaphase. This phase is defined by the alignment of all duplicated chromosomes along the metaphase plate. It's an imaginary line in the middle of the cell. It's not a real structure. All it is a reference point that's going to help organize chromosome separation. Now for the most exciting part, which is anaphase. This is when these sister chromatids are finally going to separate. The kinetochore microtubules pull them apart and drag each chromatid to opposite sides of the cell. At this point, each sister chromatid becomes its own individual chromosome. Meanwhile, other microtubules, which are our non-kinetochore or polar microtubules, they're going to push against each other to stretch and elongate the cell. Then in telophase, we're going to have new nuclei begin forming at the end of each cell. The chromosomes, they're going to begin to decondense back into chromatin, and a cleavage furrow is going to begin to form, which is where the cell starts to split. And then lastly, we have cytokinesis, which overlaps with telophase, but it is not a part of mitosis. It is a separate process that happens